So Pradeep is actually runs so he's from Blizzard Institute at Queen Mary's. He runs the QMAR epigenetics thing. I don't know if anyone sees this online, but I join all the time online, which is great. So I think I thought it would be an appropriate person to tell us a bit more about like where the histomark field is and what's coming.
Thank you very much. Thanks, Ranjan and the organizer for inviting. So I'm happy to take questions anytime. I may not go in detail certain points, but I'm really happy to discuss in detail if you want to know more or if you didn't understand. So my lab works on histone. In my PhD, I was working on the acylations and methylations of non-histone protein. And that's how I kind of got interested in more about this post-translational modification, how they can influence protein's function. So you must have come across, if you're a biologist or a chromatin or genome regulation people, so you must have come across all these different modifications on this histone tails, histone H3, H4, H2, H2, and the variants, etc. And there these various types of modifications like lysine acylations, methylation, phosphorylation, etc. And then there are enzymes there which acylates or modifies, and the one which removes. So forgetting all this complexity of so many modifications, so many, so much, code, people talk about histone modification code, and in combination they can do a lot of stuff and all that. To me, I find it too complex, and it's very difficult to understand what's actually doing each of them doing. So what I like to do is like try to understand taking one of these modification in isolation, particularly some of these modification which I think as a property with to kind of directly influence the chromatin structure, not depending on readers, not depending on the neighboring modification or anything else. So just one modification, can it affect the chromatin? So before I go into particular modification, I'll just give a quick introduction about the why people are excited about this modification, why they use it for their studies. And so one of the popular reason, like everyone studies this histone modifications in the chromatin genomics field, is because they work as a really nice predictor of regulatory elements. This is an example of so enhancer element, which has got a acylation of histone H3 lysine 27 and monomethylation. And so, and of course, they bind transcription factor. They kind of say, okay, this could be an enhancer. And then if you don't have acylation, they have a monomethylation. It could be a kind of poised enhancer or prime enhancer. It might get activated in a different cell types. So that's I think everyone, if you look at the ENCODE data on like the millions and thousands of cells, they try to profile this and predict the activity of these enhancers. So the question also comes like, what do they do? What do these histone modifications, particularly acylations, do? Like they correlate really well with the gene expression because these acylation apparently it's supposed to be reducing the positive charge of the lysine in the histone. So it kind of prevents the interaction of DNA and histone and then open up the chromatin structure. So it associates really well with the transcription. Many people believe, or kind of in the field, they have an instructive role in transcription, and when there is acylation, leads to higher transcription activity. Some of these studies kind of challenge that kind of function where you inhibit transcription, acylation disappears, and people argue it could be consequence of transcription. They may not have an instructive role. Also, some of these mutations in lysine 27, for example, or even may not be a kind of a perfect marker for identifying all these regulatory elements which everyone uses. So that's why I'm kind of going for focusing on three different modifications. One is histone H3, lysine 27, which is very well studied. Anyone's trying to map the enhancers or regulatory elements in the particular or in their favorite cell types or tissues, they use this. And also I'll be talking more about lysine 16 of histone H4. And some of the modification that's lysine acylation at histone H3 globular domain, that histone H3 lysine 122 and K64 and K56, they also occur at the globular domain of histone H3, not in the histone tail. well, I mentioned about like direct instructive role. So if you look at the literature, like there are, if you look at the early East studies, or even the in vitro biophysical reconstitution studies, for example, if you reconstitute an iron chromatin structure with modified nucleosomes, for example, if you modify lysine 16 in histone H4, it kind of prevents the interaction of this histone H4 tail with the neighboring nucleosome acidic patch of H2A. Because obviously this lysine is known to interact with the acidic patch in the neighboring nucleosome. It helps in the iron chromatin structure. It makes formation of, at least in vitro, it makes a nice compact chromatin because of this nucleosome interactions. So when you do this acylation or remove this acylation, when you make the nucleosome with acylation or a mimic, so then you perturb this kind of interaction and it loses the interaction between neighboring nucleosomes and you don't form any more iron chromatin structure. That's very well. It's beautiful in a way that you can see in the in vitro system. That in yeast, what you can do is like you can, because the histone copy numbers are like, well, not many, like one copy, so you can easily mutate one lysine in yeast and then see the effect. So if you mutate any of the histone H3 lysines, nothing happens to the yeast. The transcription is fine. They survive, and even if you induce transcription, it seems to be fine. So particularly when you mutate lysine 16, it appears that it's the rapid induction of transcription slows down. Of course, over the time, the gene expression level maintains, but if you induce a transcription, it kind of takes more time to stop. Suggesting this kind of oscillation potentially is important for rapid transcriptional induction or open chromatin. And if you remove the entire histone H3 tail, it doesn't change the chromatin structure, so that the histone tail doesn't seem to be important for the iron chromatin structure. However, there are other lysines which I said in the histone H3 globular domain, that's lysine 122, lysine 64. These lysines are present at the kind of a nucleosomal entry and exit site for this DNA, and that's where the DNA comes in contact with the lateral surface of the histone octamer. If these lysines are acylated because of the charge neutralization, these lysines no longer positive charge to attract the DNA. So it kind of leaves the nucleosome in a kind of a labile or destabilized form. It can also lead to opening of this DNA for the access of transcription factors. So this could be a direct effect of acylation. No need of any effector proteins. You don't need any BRD proteins or BRD4 to bind and recruit transcriptional activators. Well, for some of the other acylation, even including lysine 16, lysine 27, one of the commonly understood way they function is they recruit BRD4 or other BRD4 proteins, and even some of the histone acetyl transferases with the BRD bromodomains, they bind and they kind of spread the acylation around that region. So they do need effective proteins. So can we use this? Like, where are these particular modifications? Are there? My interest was like, I'm not a biophysicist anymore. During my PhD, I played around a little bit about chromatin structure, AFM, try to modify the lysines and see whether it changed the structure. It's beautiful actually. Some of the proteins, if you acylate a lysine, they're no longer able to condense the DNA. So we can visualize that it all works well in vitro. So what happens in vivo? Where are these modifications, and do they actually correlate with the accessible chromatin or open chromatin? That's what I was trying to understand over the almost 10 years now.
You're saying that, so just going back to your previous thing, and you're saying that they affect the rate of initiation. So is it the histone marks involved in just like response to environment? That's the key.
Yes, not all. Like for example, K27 may not, but not citing the paper. There's I think Rob Schneider's lab originally discovered this K122, for example. They did the again reconstitution experiment, in vitro transcription experiment, and yeast mutation experiment. Again, kind of similar to see what happens. do they behave like a K16 oscillation? Yes. So if you stimulate, like for example, if you estrogen response signaling, for example, the immediate transcription is much higher if you have these oscillation, for example, at least in the ecosystem. So we think, actually, our data suggests in this paper, we showed that many of these oscillations are there as a bivalent mark. They the regions with no K27 oscillation, but they have a K27 trimethylation, but they have this K122 oscillation. So they're kind of in a bivalent situation or a poised situation. When they get the signal for transcriptional activation or differentiation or infection or whatever, and they are able to immediately respond to that. So that's the beauty, like, why this happens, why these modifications are there when there's no oscillation, because as I said, this lysine is methylated, cannot get oscillated. The P300 still binds the same enzyme, it still binds to the region, oscillates there, keeps the chromatin in the kind of labile situation or the nucleosome. It's easy for the initial quick rapid firing of transcription.
So when you say there's multiple bivalent, do you know that they're actually present at the same locus at the same time?
So we did some kind of a rechip or whatever. Yeah, we did see in the same locus at that time. It's not a heterogeneity issue in our case, but it's also easy to believe because they basically we do see a nice P300 enrichment as well, even though there is no K27 oscillation at this site. So we see a P300. So it's easy to believe that data as well. Right. So that's what our interest was, like, to understand exactly what these modifications are. So I'll go quickly with the globular domain oscillation. As I said, at least in vitro reconstitution experiment, it affects the nucleosomal stability, and they are important for rapid activation of poised genes upon differentiation signaling. So when we perform ChIP-seq mouse embryonic stem cell, so we found these marks K122, for example, is there almost everywhere where K27 oscillation is there, like active enhancers and promoters. But we found thousands of regions where almost no detectable K27 oscillation. We found a really nice enrichment of K122 oscillation, and sometimes they kind of overlap with K4 monomethylation. This is kind of not seen all the time, and they overlap well with the transcription factor binding site and accessible chromatin as well. And some of these regions are classified as enhancers, promoters by chromatin, or sometimes they are not classified as anything. So we could show that they actually, thousands of these enhancers with no K27 oscillation can indeed act as active enhancers. They have if you perform a reporter assays, or if you delete them, or if you do CRISPR-I, you can perturb the expression of clinical genes or give some field contact. But there are something weird about these type of enhancers. Unlike typical enhancers with lysine 27 oscillation, they have a really high number of bidirectional transcription. For example, this is the group 1, which goes this way, it's a bidirectional. So the two strand is that way. So you see high level of this bidirectional transcription of these enhancers with the K27 oscillation. However, this group, the group has no K27 oscillation, but they have a K122 oscillation. So when we perform RNA-seq and analyze, they don't really show this kind of bidirectional transcription. That's a typical hallmark of enhancers. They have a bidirectional transcription from the center of the enhancers. But when we do RNA-seq in the absence of exosome, that XOSC, that's one of the genes which degrades this enhancer RNA or bidirectional transcripts of the enhancers, including some of the non-coding RNA. Their job is to basically remove all these noisy transcripts which don't code for protein. They might otherwise cause genomic instability. You don't want them to accumulate. So when we knock out that, then you start seeing this kind of accumulation of this bidirectional transcripts. So of course, the other group is basically they have no oscillation. They don't transcribe anything, even the unstable RNA. So they're different. They are not exactly same as K27 acylation, and they appear to be also some of them overlap with the K27 trimethylation. Some could be poised. So just to summarize what everyone does in the epigenomics field or someone who studies enhancers, they typically use these two marks to identify enhancers, K4 monomethylation, and they also, of course, use the accessible chromatin transcription factor binding site, and then use eRNAs as well. And they sometimes do ChIP-seq for these kind of coactivators, which does this modification, and then say, okay, this is an active enhancer because we have a K27 acylation, K4 monomethylation. And of course, in the genes promoters, you kind of see a bit of monomethylation, but more of trimethylation and dimethylation, and in association with the K27 acylation. Okay, that's everyone agrees, including me. I agree. Most of the enhancers have K27 acylation and with K4 monomethylation. But increasingly, we have been finding out there are more acylation, not just K27. There's another acylation of K9, histone H4K4, or very recently, the publication, they show that so many lysines in the H2B N-terminal lysine, they get acylated. They also typically mark enhancers. Okay, so basically, it's not just K27, there are many acylations, and enhancers are rich in acetyl marks. Okay, however, I'm not talk too much about K16 acylation. In yeast, we know really well that it's important for transcription, and then in vitro, we know that it's important for chromatin factor. In vivo, I'm not showing the data here. We did some experiment about 10 years ago in mouse ESL. We couldn't see any change in the chromatin structure when we broadly looked at the association between K16 acylation and the chromatin structure. But then again, all this data, what I'm going to show from now on is human ESLs, because the reason we used was completely different. It's about disease relevance, because many of these mutations in these genes lead to neurodevelopmental disorders. So we are about to model what happened, what's the role of these modifications or the factors in human ESL differentiation system, particularly neuronal lineage. Anyway, so we were interested in studying all of these in human embryonic stem cells. So we mapped them, and we showed that this K16 acylation marks enhancers. We published 10 years ago already. And so now we wanted to do more perturbation studies, like whether is it just a mark, do they regulate gene expression, what genes do they regulate. To our surprise, at the same time when we were studying, there were a couple of papers came. One of them is from Christian Ellen's lab. They showed that lysine 16 acylation actually is mediated by catate, of course. Catate exists in two complexes, MSL, male specific lethal, and NSL, non-specific lethal. Both same in Drosophila and in mouse or human. The complex is highly conserved. They do the exactly same thing. The catate, when it's in MSL complex, it does the K16 acylation. Catate, when it's in its NSL complex, acylate H4K18 or H4K5 lysines. These lysine acylations are enriched at the transcription start site, and they associate with the active genes, so promote the transcription. When you perturb this complex, or when you delete any of these, basically cells die. They are essential genes. You can't really make the knockout of this. However, this K16 acylation and also MSL complex seems to be not really essential complex in a way. It's important for development, but also this K16 acylation doesn't mark at the promoter or the transcription subsite. Their publication, they showed it's very broad mark, which marks the A compartment, not the B compartment. It's widespread, and although there's no mechanism like why it's spread out in the entire A complex or A compartment, or what region do they mark?
Say what the A and B compartments are.
Oh, sorry. I thought Shinjan has introduced that a few days ago. So basically A and B, if you divide the entire genome, I think now there's a D compartment as well. So if you divide the entire genome into two, it may correct me, Shinjan, I'm not a 3D guy. So you broadly categorize them into active region of the genome where there are a lot of genes, more gene expression, a lot of euchromatic marks, and
Yeah.
and the B is some kind of heterochromatin silence.
But it's not euchromatin heterochromatin.
No, it's like H4 contact.
Yeah,
So
it's
they
based
take all
on the
H4 contact, split all the H4 contact into two compartments, and you get A, which is mostly active fields, mostly. And then recently, obviously, sequence it deeper, and then you can split it into more things. So now you have A1 and A2 and B1 and B2, and then
yesterday there's a D compartment
D,
yesterday.
like now there's all these compartments. Well, obviously, if you sequence deeper and deeper, you're gonna split it into more and more compartments.
Yeah, so it's a very broad. I think they are in the megabase, I think, scale in this. Yeah, it's not a gene. There may be genes in the B compartment which are expressed particular lineage, right? Yeah. So anyway, so it kind of, that's what they argued, that it just marked the overall expression. The same year, one of our collaborators from Crick lab, they were studying the same thing in cancers. They did a CRISPR screening, and they found, again, kind of similar. The MSL complex is kind of not really essential, but when you knock out this, so you don't see a changes in gene expression. Instead, you see a accumulation of genomic instability, and cells become aneuploid and then collapse at some stage. But there's no change in gene expression. So if you remember, I argued that K16 is very important mark in vitro. It's beautiful, it opens the chromatin. You don't see any chromatin if you have a mutation, and yeast, it works perfectly. And when it comes to IR organism, you don't see that. You don't see a gene expression change. So we routinely use, I think Shenzhen knows, we use this method called cut and tag. So one of the very simple way to profile a histone modification. You can do, like previously ChIP-seq was kind of, people are doing few, but you can do hundreds of them in a matter of weeks. This experiment basically, I don't want to go too much into detail. Basically, it's a transposase-based method where you incubate primary antibody and then add a secondary antibody, and then you fuse an enzyme transposase which binds to the secondary antibody and primary and adds an adapter, and then you basically make a library and sequence it. It works beautifully for us. The reason I'm saying this is because this makes a big difference for our data. Previously, the data which I mentioned from Christian Allen's lab, it was very broad. They couldn't see a really nice peak. They kind of marking the entire A compartment, the K16. Now we could see, like, it's not really the entire A compartment is marked by K16. We could see a lot more details, like, for example, the gene bodies or towards the end of the genes have a K16. Also, some of these promoters, but not all. And some of the enhanced elements also have a K16 oscillation. And we compared with many other modifications. So, and then we did something which everyone does, like, just to blindly see where does these modifications go, how do they compare in the genome. When we do this Chrome HMM, hidden Markov model of identifying which segment these modifications are enriched, to our surprise, we find a lot of K16 oscillation in the heterochromatin and the insulator region and the repetitive elements. So much higher than the other modifications which are known to be enriched in the regular elements. It's kind of K16 oscillation was kind of looking similar to K9 methylation. This is known to be a proper heterochromatic mark, which is enriched on the transposable element, which is tightly associated with the heterochromatin. We are shocked, why do we have K16 oscillation? In vitro works beautifully for the active chromatin or the open chromatin. Why is it going to heterochromatin? So then we had to analyze that in deeper, like then before I go further to our analysis, I just have to introduce about this genome again. Our genome, nearly 40 to 45% is kind of made up of transposable elements of different families. One of them is line 1, SINE, endogenous retroviruses, and a few other like LTR-based elements, and of course the genes. And when I say genes, that's only exons. Of course, there will be a lot of introns. Introns will be everywhere. There are a lot of these repeat elements have introns in them. So if you just look at the genes, they are only 1%. And if you look at the promoter, it will be even smaller fraction. and rest you just ignore the rest of the genome for now. So the thing is, you got nearly 40% of the genome transposable. Some of the transposable elements, which they don't do anything. Basically, they lost all the ability to recruit transcription factors. They lost ability to transcribe. They lost ability to retrotranspose. They're just a piece of DNA there. Remove all of them. You still have 10,000 of line 1 elements. That's a long interstitial nuclear element. So basically, they are about 5 to 6 kb long, and there are 10,000 of them. They potentially can still be transcribed, although very few are known to be retrotransposed. And they retain the transcription factor binding site. They act almost like a gene, except the introns. And so that's a structure. Like basically, it's like a promoter, and they code for open reading frame and another open reading proteins. And these proteins are important for the retrotranscription. And also there are nearly 1 million sign allele elements, and there are hundreds of thousands of endogenous retrovirus. Not all of them again full length. Some of them are truncated. They only retain long-terminal repeats. These long-terminal repeats again can bind transcription factors and activate transcription or influence the chromatin. Okay, why did I say that? So of course, these are there in our body, but they're all kind of tamed or silenced by various transcriptional repression machineries or post-transcriptional silencing machineries. So even epigenetic factors and also accumulation of mutation over the generation. So basically, they are getting silenced or not being active anymore. So I don't want to go to any of the repressive mechanism now. My talk is more about activation or euchromatin now. Okay. So yeah.
So I'm not sure whether I understand this post-transcriptional repression in context to your other what you said earlier. So maybe,
yeah. So maybe I don't understand as well. So this is something maybe Eric Miscavin. Yeah. So this is something like, once, like, imagine you have a transcription, there's a leaky transcript coming from these elements, even though all this, we have a DNA methylation or this H complex are said to be one, and all these things are trying to repress this transposable element. They can still escape this repression, and they can still come out. And then, and also these can be tissue specific, particularly in germ cells. They are kind of, this is one of the major mechanisms. These transposable elements are silenced. So each cell types, each tissue types potentially have a different, also depending on the type of transposable element, they use combination of these machineries to repress or reduce the expression of this transposable element. So the pdRNA or like pRNA or PD complex, basically again, they degrade any Ts which are expressed, which are, even though they are supposed to be repressed, but not all are repressed. So they are taken care by a post-transcriptional mechanism. Right. As I said, you've got this millions of these elements, and they all, many of them still have a potential transcription factor by inside. They can recruit T transcription factors. Imagine if they are present. So before that, I need to kind of say the importance of this. Say the presence of these transposable elements in the gene body or near the gene or near or in the 3D contacts of this transposable element with the gene can actually perturb the transcription of gene of interest, because they are in the same location in a linear space or even in the 3D space, can actually influence each other's expression. So we don't want to have a kind of environment where the entire 50% of the genome is buried with these repressive elements, and there's if there's any particular genes are there or these elements are retrotransposed near the genes, and they kind of create a repressive environment. So I think where I'm going from there is basically to argue that some of the elements, some of these transposable elements, they still retain the ability to bind the transcription factors. They still retain the ability to transcribe, at least in the basal level. That may be still useful for our genome or gene regulation, because they might be used by the genes nearby for their own benefit. For example, nearly 20% of the transcription factor binding sites are within the transposable element. And and then, for example, there's many of them are tissue specific. For example, if you analyze all the transcription factor binding site in the different cell types, and then if you look at the tissue specific transcription factor binding site, many of them fall from the transposable element. This is just an example, some of these transposable elements, which are specific to particular cell types. That's early development, neural crest, or placenta, or neuronal lineage elements, and the transcription factors, which are specifically bound to these transposable elements, and they can potentially regulate the expression of these transposable elements. So the reason I showed these three major types of tissues is because we know that in early development, this transposable element plays a key role. Again, they are highly expressed. They might be involved in even for the there are reports that they're important for 2-cell stage and other things. And in neural crest, because again, some of these transposable elements are particularly transcribed in a higher level in the neuronal lineage. And in placenta, I was about to present this, but maybe for other way. Placenta again have got many transposable elements which are expressed particularly at the placental tissue. Indeed, some of the genes in placenta are derived from the transposable element.
So you're arguing that these transposons actually have to be activated at certain points of development to correctly develop to happen.
Yes.
So what are these transposons actually doing? They're acting as transcription factors themselves.
So the one example I'll give you, I removed the slide a lot, and it is a gene called syncytin. So the syncytin is a kind of derived from a viral envelope protein. In placenta, the placentation to occur, the trophoblast stem cell has to be differentiated into two lineages, syncytial trophoblast. And basically the syncytial trophoblast function is to fuse the cells. The gene N from a virus has been evolved as a placental gene. It helps in fusion of cells, and then that's how the placentation plays a major role in the placentation. I think human placenta is completely different. It's also very unique to human placentation. So it's complete different type of evolution, not a transcription factor, but as a cell fusion role. But some of them, they don't act as a transcription factor themselves, but they recruit transcription factor for the way, say, for example, the virus need to be transcribed and replicated and spread across. So they retain this binding transcription factor binding site because they're supposed to be mutated by our genome, and we don't want to keep the transcription factor binding site, but they do, many of them, they still retain this binding site. So these are the transcription factors, binds to the transposable elements, and then activate the transposable element, tissue specific way. And these transposable elements have a tissue specific function. What is the function? One example I gave you, there are few genes like the placental specific, and there are also another way they can function. I think rest of the talk will cover that. Yeah.
I think that's why I get worried about my nano. Because nano is like when it gets recruited to all of these transposons. It could be that because we see this clustering of nano, and it could just be because there's like a lot of binding sites in some of these.
Stem cells do express a lot of transposons.
I just assume the transposons have side.
Many of them. Because there's so many of them, because people ignored all these years. We were ignoring because they're tricky to map, and we all just go for this 20,000-30,000 genes or non-coding RNA, which are typical, and then analyze to make our life easy.
mean, I thought transposons only had to be activated in so much they had to be activated and silenced by, by RNAi and stuff like that.
No.
But this is something quite different.
Yeah, there are multiple ways they can be silenced. Yeah. They don't even need to be silenced because they can accumulate mutations, so they won't even recruit transcription factor, some of them. But if they recruit transcription factor, they're more likely to be transcribed, at least in a basal level. So yeah, coming to our own data again. So what we expect K16 acylation to be enriched at the genes and the transcription start site, and maybe a bit on the gene body, because it's kind of known that in K16 acylation is enriched on the gene body. It's important for transcription elongation, at least in Drosophila studies. To our surprise, we find really good enrichment of this K16 acylation at the line 1 element, even the K122, but not so much of K27. There's some K27 as well. Almost really as good as K9 methylation, which is known to be enriched at the transport element. Okay.
So just go back one. So that line 1 class, it has got K9 trimethyl and K16. So it's kind of a transposon bivalence or something.
Yeah. Absolutely. The next slide covers that. So you expect all these transposable elements with the K9 methylation to be silenced, not expressed, and it's taking care of its repression role. But we see this acylation there. And what is this acylation doing? So if you look at the gene, for example, so this is the gene transcription start site here and end site there. And you see K4 monomethylation at the gene promoters, and these are just three replicates. You can look at the one replicate. And not much K9 methylation in human ES cells. There may be few genes have a K9 methylation, not much. And you have K27 acylation as you expect at the promoter. And bit of K122 at the genes. And the K16, as I said, it's on the gene body, a little bit toward the end of the gene, tiny bit at the promoter. Okay. If you look at these line ones now, this is the line 1 I mentioned before. This is the kind of promoter of line 1, 5' UTR, right, transcribed bidirectionally, and also this is the promoter. So you see enrichment of this K16 acylation, the promoter of this line 1 element. So at K12 acylation. A tiny bit, some of them have a K27 acylation, but K27, as you know, it's more of an answer and promoter, not much. But we have a really good K4 monomethylation at these line 1 elements. And not much K4 trimethylation, because this is more of a gene promoter. So it's really intriguing. Like, it looks like an answer because there's K4 monomethylation, there's some K16 acylation, which we know that this can mark an answer. K122, I've shown that it also marks an answer. So what is it doing there? When we look carefully, some of these elements. So many of these are all full length. There are about 10,000 line 1 elements here. So they are classified as the line 1 human specific, line 1 primate specific, L1 PA is basically L1 primate specific. So about 100 million year old in the primate genome. This is younger, PA2 is younger than this one, so they are different time point. They have been accumulated. So this is a 5' UTR of this line 1. You have a really good K122 acylation, K16 acylation, not much of K27, and there's a K4 monomethylation, while this one has almost no acylation whatsoever. So let's just to give an example. So you mentioned about the bivalency, K9ME3 and K16. So this is the K9ME3 on the line 1s, different evolutionary time scale. These are youngest line 1, human specific. They are not there in primates or any other organism. These are primate specific. These are older. These are kind of mammalian specific. Interestingly, some of these seems to be regulated by different mechanisms, because we know that if you look at the DNA methylation, each of these line 1s have a different DNA methylation profile. Some of them may be repressed by DNA methylation, some by K9 methylation, different mechanisms. So K9 methylation, obviously, it seems to be there in kind of a line 1 primate specific PA3 to here PA6. While they don't have much K4 trimethylation, there's some. And some of them have a K4 monomethylation, almost all have a very low level of K4 mono. K16 seems to be enriched in the older line and some of the younger line variants. And many of them have an accessible chromatin as measured by ataxi. Some do by BRD4 as well. So they all kind of shows the signature of some of them are repressed, some could be
So isn't that strange that in the lowest one, they've got high levels of K16 acethyl, which should be opening up the chromatin, but by ataxi, they've got the lowest accessibility.
Yeah, that's exactly what I was trying to say about K16 and that's ataxi. I forgot to detail one of the previous study, Christian Allen's lab. They did ataxi upon knocking down or reducing the K16. They didn't see much changes in ataxic signal, but again, they looked at the genes. So I think that I don't think we have very strong evidence K16 plays a major role in opening up the chromatin, at least in the cells. In vitro, it works beautifully, but not in cells. We don't know. So, but it does something. When you perturb, when we knock out K16 and look carefully where the K16 binding is there, hopefully I'll show in a couple of slides. There is some change.
But is ataxi really measuring? It's measuring chromatin accessibility.
Not the structure. Yeah.
As you know,
Exactly. It's not exactly what it's measuring. Yeah, you're right. Okay, and also these elements appears to transcribe when we use them in a reporter system, like it's a star-seq it's basically you clone all these entire genomic region and then transfer it back to the cells, and then I'm sure Stefan must have spoken about. Then you start measuring whether the elements can drive the transcription of a reporter system, like it's kind of a in vitro assay. So very strong or kind of more powerful in a way. You can study the entire genome, which part of the genome has a reporter, like an answer-like activity. So we can see very specifically this 5' UTR can function as an answer, at least in the in vitro report process. That's again, I spoke about line 1. So these are some of the classes of LTR elements, that's ERB LTRs. They also seem to have some of them K16. Some have a K4 mono and K27 as well, but that is of K16 mark for LTRs and no K27, right? And when we analyze the star-seq data, very interesting observation here. I have no idea why. I mentioned about K27 oscillation being a widely used enhancer mark. When we look at the enhancer activity in the genome, okay, wherever there is a K16 oscillation along with K4 monomethylation, we see highest enhancer activity. This is again focusing on LTRs. The LTRs, that's transposable elements having K4 monomethylation and K16 have the higher level of enhancer activity compared to just K4 monomethylation or K16 oscillation, but lack K4 monomethylation. They also don't have much. But when we add K27 oscillation to the same regions, for example, some may be different, obviously, they are not the same regions. There will be, when we pull out more regions with K27 oscillation, so it doesn't increase, rather it reduces the little bit reduction in activity. I think that difference is much more stronger here. If you look at the K16 alone, enhancers have a higher star-seq activity, but when we add a K27 oscillation, you have a reduced enhancer activity. No idea why. Something it agrees with some of the papers. They show that K27 oscillation, which is widely used as active enhancer mark, doesn't show higher enhancer activity in the reporters. I don't know why. It's very, very weird. Yeah. But anyway, so this transposable element, as I said, have various oscillations. They bind to transcription factors. Interestingly, many of them are enriched for CTCF, YY1, RAT21, particularly line ones, and LTRs, some of them, for example, are enriched with F143. They are important for these bold. The one in bold are important for higher order chromatin structure and looping and so on. So this is how it looks. For example, in the LTRs, you see really good enrichment of RAT21 CTCF and YY1, and the line 1 again in the 5' ETR. You see a really good YY1 enrichment. There are 2 YY1 binding sites. You can see really nice 2 enrichments peaks there, and a bit of RAT21. CTCF is a bit low, but still has some specific enrichment. And we show that these LTRs or ALUs or line 1 appear to loop with the genes when they are oscillated, compared to when there is no oscillations. This is line 1s with no oscillation, line 1s with different oscillation. They more tend to loop with the genes. It's same with all 3 classes. I think there are more evidence with ALU. Recently, there are a few big papers claiming that because of ALUs or kind of repetitive nature, they tend to form kind of a hybridization, and they make a very strong looping interaction with analysis in the genes there because of base pairing. No, basically what we did is like we took all the LTR or all ALU elements, group them into whether they have peaks for the oscillation, particular oscillation marks, or no oscillations in them. So the one which no oscillation, none of these oscillations tend to not loop with any of the genes. So what do they mean? Like now, for example, if these transposable elements are there everywhere, they're 40–50% of the genome, and of course not all of them are oscillated. And when we look at only the transposable element, for example, line 1s with the K16 oscillation, so what happens to the genes which are located near the transposable element? So the genes less than 10 kb from the transposable element seems to have higher expression level compared to the genes which are away from the transposable element. Okay, so that's only very specific to K16 positive, and K16 negative line 1s, they show the opposite trend. So basically showing that the presence of this K16 oscillation or the transposable element with the K16 oscillation somehow influencing the expression of genes, or at least they correlate with the gene expression level. So that's line 1. It's a similar story for LTR. When they are closer, more likely to be highly expressed. When they are far away, when the genes are far away from these LTR, they are less expression. So it's all nice and correlative. Do they actually do anything? So what we did, like now, of course, everyone uses CRISPR now to perturb elements. So we did 2 approach, CRISPR-I, basically we recruited CRISPR-Cas9 with the craft domain to the transposable element, some of them with oscillation, some of them with no oscillations, for example, but they're all located upstream of this gene, ESP38. So I'm just showing one gene, but we have done a few others, but I think it's easier to explain this one. When we do CRISPR-I for this and this, there's no change in expression of ESP38. And when you do CRISPR-I for this line 1 and this line 1, it affects the expression of ESP38. Look at the distance, they're about, I don't know, 50 kb away or 50 to 100 kb away from the gene. You can still affect the expression of gene nearby. But it's very specific, it doesn't affect the genes nearby, they don't make a looping interaction. That's another example of line 1, which is there in front of this MOXP1 gene. It's again very big gene, 100 kb, where it loops and make a very significant interaction with this gene 5' and also gene very next to that. It doesn't loop this one. So when we do CRISPR-I, it perturbs the expression of MOXP1 and also STX7, but not ENP1. I mentioned about CRISPR-I. So we did CRISPR deletion as well. We basically deleted this region, used 2 guides to delete from here to there. It's tricky to delete only this part because of the repetitive nature. So we have to delete the entire 7 kb. So before I say that, yeah, we also deleted this line 1 and this line 1 as well. I'll show you the data, what happened. So that's the CRISPR-I. When we did CRISPR-I, for MOXP1, as I said, it reduces the expression of this gene and this gene, but not the ENPP1. And when we did CRISPR-I for the 2 one of these enhancers. So it expresses the it reduces the expression of USP38, but not other genes nearby. Also, when we do CRISPR-I for the other K16AC negative line 1 elements, although these are closer to the gene, they don't express the USP38, so they don't affect the USP38. So this one is very close to this gene compared to this and this, still no effect on expression of USP38.
It does not affect the loop.
We didn't study the looping. So the looping also is not, we don't, this is kind of a RAT21 iChip data, bit noisy. I don't know whether you call it a loop, but they are in a same 3D space, I would call. This one is a clear loop with a very statistical enrichment. Okay, so not showing too much in because very interesting. Like there's in a way, if we delete, we have a slightly different result, but I think that could be due to CRISPR-I being much more stronger effect than the deletion. Anyway, so thing is, many of these oscillations, they're tricky to study in vivo in the cell culture model or even in the cell culture model, because if you delete P300 or any acetyl transferase, hoping that you want to study the role of K27 oscillation by removing the P300, you perturb many more oscillation, not just P300. You basically, because many of these acetyl transferases, right, oscillate multiple lysine genes. But the beauty of this K16 is it's only mediated by cat8, that too only when it's in the presence of MSL2, 1, 2, and 3. So if you knock down or knock out these 3 proteins or one of these 3 proteins, you lose completely K16 oscillation without perturbing even the other oscillation mediated by the same enzyme or K27 oscillation by other enzyme. So, so that's what we did. We basically did the knockdown and knockout. This is just knockdown data. When we knock down, we could see that there's a reduced chromatin, like an FX6 signal in the K16 depleted regions. Like this is where the K16 enrichment. And when you look carefully, some they don't show any changes, but some reduce the accessibility. If you look at them carefully, they all seems to be younger line 1 elements. And also these are the ones which shows the reduced transcription when you compare to control. Upon depletion of K16 with this enzyme, you see reduced transcription. So this is just a knockout, just to complement the data. So for example, you can do CRISPR, you use a CRISPR to knock out MSL, and then you use the K16 level, that's the magenta is the K16, you use that, and you also lose the line 1 open reading frame, that's one of the proteins produced from the line 1. So you basically lose you can quantify thousands of cells and then show that, in the absence of line 1, in the absence of K16 oscillation, we don't see transcription of this transportable elements. And also it reduces the chromatin accessibility, very subtle. It's not a huge difference, but it seems to be very specific here. And the genes, it doesn't change much. As I said, the genes. Yeah, I think it's almost end. So we basically the same thing with the knockdown, and we see the same reduction as well. So I mentioned about ALU elements. We don't really see enrichment of any of the typical enhancer features like ataxic signal or K4 monomethylation. Some oscillation of K16 and K122. We kind of ruled out in our publication that they in this cell type they may not do much as an enhancer activity, but they seem to do something different because they express repetitive RNA. They can complement each other, like the alleles are present near the enhancer and promoter, they can loop and they can form this kind of looping interaction. Very interesting observation there. Okay, just to summarize the working model. So we have transposable elements everywhere. They are in the introns. They are located at the enhancer element or the themselves acts as enhancer element. And presence of these repressive modifications like K9, ME3 or DNA methylation can influence the expression of genes which are located very close to these transposable elements in linear distance or in the 3D space. And sorry, this came as a extra thing, but we show that K16 methylation can create a kind of a active mark and also regulate the expression of this transposable element, and then and which can influence the expression of genes nearby. Also, so many of these transposable elements are known to, because they are viral DNA, the cells can sense them as a viral nucleic acid, whether it's RNA or DNA or cDNA, and then trigger the interferon signaling as an antiviral response. It's kind of implicated in many disorders as well, like the upregulation or dysregulation of transposable element can lead to kind of a sterile inflammation. Okay, I think that's it. Happy to take more questions, I guess. So I think we are doing that experiment now. So the whole idea of the project was to make it neuronal differentiation and whatever, but we got stuck with so much data, and I think Srinjan knows that we didn't have enough resource to go into differentiation, but we are now doing it.